proteins are my favorite. Pretty much we are expression of proteins that are present in our trying to change my highlighters. Okay, so we are our expression, whether it's our skin, our facial expression, our hair, they're all composed of proteins. And here we have shown you three images or three different kinds of ways that the proteins can form chains or organize themselves into more complex nature. And I'll give you more examples about these later. Proteins are my favorite because proteins have numerous functions. Some proteins are for structural support. For examples, proteins that are present in the cell membrane. They will give you structural support. They are present. Certain proteins are only for enzymes, that is, they catalyze reaction of all sorts within the cells. Transport protein, for example, hemoglobin, it's going to transport oxygen in the blood. Defense, example, antibodies that are created or antigens even that are formed, and these are defense. Hormones are also proteins, for example, insulin. And motion, for example, actin and myosin fibers that are present. So, and structural will be, let's support could be, for example, in hair and nail and hooves. Those are structural proteins. So you can see that they have a wide variety of functions, and that's why proteins are almost essential macromolecules. Proteins are also called as polypeptide. So these are long chains of polypeptide. A polypeptide is a long chain of a simple unit, which unit is shown in a representation in each of these balls, and these are the monomers. And the monomer in general for proteins are called as amino acids. So let's go on to look at more about the amino acids. There are firstly only 20 common amino acids, and there's a diverse expression of proteins in animals and plants. They all have a common structure, and the common structure of protein is represented over here. You write down the central carbon, you put 4 bonds. The bottom is an R group, which means it could be any number of carbons, a hydrogen on this end, and there is a group that is called as a carboxyl group on one end, and another group which is called as the amine group on the other end. So this is my amine end, and this end is my carboxyl end. Now these are functional groups that I mentioned earlier. They are again joined by the same dehydration synthesis, and you again notice the same pattern, that the water molecule is released when two monomers are put together. The bond that they form are called as the peptide bonds. So together via, as a matter of fact, that shouldn't be it, together via dehydration synthesis, we are able to make peptide bonds. Very important to remember the bonds for each monomer that is forming a macromolecule. So let's look at the different shapes of the different ways these proteins can organize themselves, and the shape determines its function. There are 4 levels of this organization. The first level is called as primary level, and in the primary level you see that amino acids are linked as looks like a bead, so they're just linked up in a linear pattern. This is called as a primary structure. In a secondary structure, the long chain is either folded, for example in here you can see the folds like an accordion, or they are wound like helical structure. and the helical structure reminds me of a telephone cord. So these are secondary structures. The tertiary structure is the one that will form by additional folds of a secondary structure. So after the secondary structure has formed and after this protein has folded in this form, you can have additional bonds or linkages formed between side groups, and they will form the tertiary structure. And eventually the folds will then form more complex structure, which is the quaternary structures. And in quaternary structures, usually you have two polypeptide chains. So imagine giving a cat two yarns to play with, and that's how a quaternary structure, the big knot that you will get. Now at this stage, it is also important to know the different bonds that help form each structure. The primary bonds are usually formed because of peptide linkages. So peptide links will give you the primary bond. Secondary bonds are usually by hydrogen bonds. Remember, we did hydrogen bonds before. So hydrogen bonds will help form, you can see over here all these hydrogen bonds are forming, and these will give you the secondary structure. The tertiary structures are other bonds besides these two, and there are all the other kinds of bond: polar, covalent, non-polar, disulfide bonds, and so forth. The names that I'm not going to go into more depth for you, but additional bonds are needed to compact or organize this long chain of peptide into a more condensed form. Now, some other properties of proteins. Some proteins are fibrous, which means that they are either structural, and these are going to be hydrophobic. Structural proteins are going to be present on your nails or your skin or your hair, and these are giving you the structural appearance, and they're definitely not water soluble. However, the globular proteins are water soluble, and these are your enzymes, and they are called as the hydrophilic. So these are just other classes of proteins, or you can classify all kinds of protein in the either fibrous proteins or globular proteins. And the most simplest way of saying that is globular proteins are water soluble. So if you have water soluble or hydrophilic, then they are globular proteins. Other than that, it would be fibrous. So think about the albumin in the egg. What do you think that would be? Can it dissolve in water? If it does, then it's going to be a globular protein. Another kind of proteins that are lately getting a lot of attention are called as the chaperone proteins. And chaperone proteins are proteins that help guide organization of other macromolecules. And prions are an example of chaperone proteins, and mad cow disease, which results to a malfunction of these, is an example of prions. The last thing I want to do before we complete the section of proteins are easily denatured. Denatured means it can be destroyed. So imagine taking an egg and either frying it or having a boiled egg or poached egg and trying to turn it back. You really cannot do it. Why? Because heat destroyed the protein. Heat, besides heat, another agent that destroys proteins are pH. So sudden changes in pH will also disorganize the quaternary or the tertiary structure of proteins. And the structures of proteins are destroyed. The protein is normally not functional. Here's an image of enzymes, and enzymes are protein in nature. So one of the most important function of the proteins is enzymatic, and you can see the substrate being attached over here on a region, and this region is called as the active site. The active site is a region in which there is a unique configuration over here. I'm trying to get my highlighter. An active site is a region which has the same configuration to a substrate, and if you have the right substrate, then it can fit in the active site and cause changes within the molecule. The enzyme concept about the active site is discussed in your book, and it's called as the lock and key model. Lock and key model is simply in stating like if you have your own lock, then you can open your own car. Same way, an enzyme is very unique and it will only function to the substrate that can fit into its active site. So an enzyme with its true substrate will form an enzyme substrate complex, and then there will be some configurational changes within the substrate, and it can either release product and the enzyme is released free. This enzyme is used back in this reaction. We'll talk about this enzyme later on.